What do sickle cell anemia, cancer-causing p53 mutations, and ALS-causing SOD1 mutations have in common? They're all caused by a single incorrect letter in the genetic code. Wouldn't it be nice if there were some way to edit genes? Enter CRISPR, or more specifically CRISPR-Cas9. It's an enzyme that's well known for its ability to quickly and accurately find a very specific DNA sequence and cut it. Cas9 uses a strand of RNA called the guide RNA and unwinds double-stranded DNA to see if the DNA is able to base pair with the guide RNA. If it is, snip. However, normally, the Cas9 enzyme makes a double-stranded break in the DNA, usually rendering the chunk of DNA useless instead of correcting it. Even with technologies like homology-directed repair, which floods the cell with the correct genetic template, the rate of correcting the gene is low. So we have a single letter in the genetic code that needs fixing. What do we do? The first step is to not use Cas9 as a nuclease, or enzyme meant to cut DNA. By disabling both of the cutting domains on the Cas9 protein, Cas9 turns into catalytically dead Cas9, or dCas9, which can still unwind DNA and attach itself to a specific sequence of DNA base pairs, but is unable to make any cuts. Wait, how does this help? This is where the next step comes in. There are several enzymes able to change one base to another in DNA. For example, cytidine deaminase, which changes a C in the genetic code to a U, which is effectively a T. However, cytidine deaminase enzymes only work on RNA or single-stranded DNA, and in the body, all DNA is stored as double-stranded DNA. This third step comes from combining the two. Remember how Cas9 unwinds DNA? This effectively turns both sides of the unwinded strand into a single-stranded DNA temporarily. By attaching the cytidine deaminase to the catalytically dead Cas9, when the Cas9 attaches itself to a strand of DNA, the cytidine deaminase can make edits on the unwinded section. Now you get the benefits of both Cas9 and the cytidine deaminase. The dCas9 finds and attaches itself to a specific sequence of DNA that you want to target, and the cytidine deaminase is able to change just one single letter. Base editing is a powerful technology and just one of the many examples of how CRISPR can be used in both the research and cure of genetic diseases.